So yes, today I will be talking about how plasticity shapes triplet connectivity motifs in spiking networks. Sorry, just a second, I lost my screen. Okay. But yeah, before getting into plasticity and how it changes connections, I would like to take a quick step back and look at how we can describe structure in the brain. And one useful way to do that is to think of the brain as a graph where the nodes are neurons or the edges are the synapses. And from this point of view, we can study the connectivity at different scales. For example, as we just saw in the previous talks, we can study connectivity at a global scale, and where we can see large-scale patterns such as sympher chains or assemblies, where groups of neurons connect in specific ways to support different functions, as Christoph just explained. But we can also zoom in and look at local structures, and here we can focus on just a few neurons and analyze how they are connected to each other. Therefore, taking into account like just a few neurons and also the direction of connections that they form, and so treating the circuit as a directed graph. These small connectivity patterns are called motifs, and this is what I want to focus on in my work. And so given a neural network, we can look at motifs within that network, and since the number of possible motifs can grow exponentially with the number of neurons involved in the motif, we typically focus on 3 neuron motifs or triplets because they are the simplest but still very informative local structures. And we know these from experiments across different species and different brain regions. And what we know is that even if they involve only a few neurons, motifs can give us important insights about how connectivity is organized in the brain. For example, already 20 years ago, Song et al. Quantified the motif frequencies in the rat visual cortex and then compared those values to a completely random network, so to the frequencies that we would expect in a fully random network. And in this bar plot, you can see how the real data deviate strongly from randomness. And another example is a more recent study from Turner et al. In 2022, where they used the microns dataset, so connectomic data on the mouse visual cortex, and this bar plot shows a similar result. That is, the brain connectivity is far from random. But also, what we can notice is that the distributions are not the same, so there is also heterogeneity in the motif distributions. And in our work, we wanted to understand how these specific motifs emerge through plasticity. So exactly how do triplet motifs form? So given this question, to understand this, we need to think, first of all, about the continuous feedback loop between connectivity and activity in spiking networks. So we can take again our recurrent network, and we can start with how connectivity influences activity. And in particular, we can focus on the activity of a single neuron, which I will call neuron I. And as we can see, the neuron receives many inputs from many other neurons, and we can see a zoom here. And the activity of neuron I, we model the activity with 2 components, which are baseline intensity, which we model as a Poisson process. And then we take a weighted sum of presynaptic inputs, where each input is a smoothed spike trace of the presynaptic activity, which we scale by the synaptic weight. So this process was introduced in the 70s by Hawkes, and it's very useful to capture the reciprocal influence of the spiking neurons in a recurrent network. And to see how connectivity exactly influences activity, we can consider, for example, the connection from the red neuron to the postsynaptic neuron I. And we see that if there is a low weight from the red to I, then the activity of the postsynaptic neuron will be barely affected by the red neuron. For example, even if there is a high activity here, this doesn't necessarily is not necessarily reflected in the postsynaptic neuron. But if the connection is strong, then the neuron I will be strongly driven by the red neuron. So this is one way to see how connectivity shapes activity in a recurrent network, but this relationship also, as I said already, also works the other way around. So activity can change connectivity, and this brings us to plasticity. And here I want to take into account the spike timing independent plasticity, or STDP, which we already heard about in previous talks. But it's a simple rule which describes how the precise timing between presynaptic and postsynaptic spikes determine whether a synapse gets stronger or weaker over time. So given pre- and postsynaptic spike trains, we can write the synaptic change over time as a function of these spike trains and an STDP kernel, which we call here L of tau. So usually this kernel L is chosen to be the classic Hebbian rule, where we know, or most of us already know, that if a presynaptic spike occurs before postsynaptic spikes, then we fall in the blue region here, denoted by LTP, which stands for long-term potentiation, so the weight gets stronger. And if, on the other side, if the presynaptic neuron comes after the postsynaptic spike, then we fall in the red region, LTD, which stands for long-term depression. So the weight gets weaker. But this is actually just one example of the kernel of a plasticity rule, because experiments have shown that STDP can take many forms. Many different kernels can model STDP based on the type of neuron, or the type of synapses, or depending also on the neuromodulators involved in the plasticity mechanisms. So here I will classify kernels into three big families, which are Hebbian, as we just saw, anti-Hebbian rules, and symmetric rules, which are all different types of STDP rule, where we get always potentiation or depression depending on the reciprocal timing between pre- and postsynaptic spikes. So this is how we see the way how activity shapes connectivity. And just to summarize this first framework, on one side we have neural dynamics modeled as Hawk's processes, and on the other side we have synaptic plasticity modeled with STDP rules. And this framework, when we put these things together, has been used also in previous work, and we can also add a third hypothesis, which is the separation of timescales. So namely, the synaptic plasticity occurs much slower than neural dynamics, which are faster, much faster. And in this framework, we can actually compute the averaged weight evolution over time analytically through a simple equation, which depends on firing rate, that we are calling R here, and then connectivity matrix. But what we want to focus on in this work is these coefficient M's, which are directly linked to the STDP kernel. So to see this, basically, if I change the kernel function here, I will change the coefficient M's, and therefore also the synaptic evolution and the final weight outcome. So, for example, I can change between Hebbian, anti-Hebbian, and symmetric shapes, but beside switching between families of rules, I also want to modulate the parameters of the STDP rule, because again, neuromodulation can shape these functions. And this plot shown here shows an STDP which is perfectly balanced. So I'm taking the ratio between LTP and LTD, and in this case the two areas are equal. And I'm also taking the ratio between the time constants of depression and potentiation, and they are also equal. But I can, again, I want to modulate these parameters, and so, for example, the kernel will take all these different forms. And the same holds for the anti-Hebbian and the symmetric rule. So given this variety of kernels of STDP rules, I want to know which motifs emerge through a single STDP from each of these rules. And since we have an equation and this problem is analytically tractable, we wanted to predict which motifs can form, triplet motifs can form for starting with Hebbian rules and starting with a simple system of only 3 neurons, 3 excitatory neurons. And what we could do is to find an analytical mapping between these parameters of the STDP rules and the possible classes of motifs that emerge. So, for example, when LTP was much stronger than LTD, as we would expect, the triplet motif that emerges was the all-to-all connected motifs. And on the other side, when LTD is much stronger, then we get the non-connected motifs. In between, more interesting cases, which I classify here below in the legend, and we have, like, for example, the orange classes, which allows for no bidirectional connections, and then the green region, and the yellow region, which is an intersection, can be seen as an intersection of the two. The white region is a region where, in principle, any motifs, we would predict that any motifs can emerge, but actually, the dynamics of the neurons will push the region into a green or yellow motifs, and this was confirmed also from numerical results, as we could see here, for example. And the rest of the parameter space also matched the analytical prediction. So the Hebbian rule could give rise to many motifs, but we couldn't cover the whole set of motifs, so we investigated which motifs emerge through the other families of STDP, so the anti-Hebbian rule. And what we observed was that the anti-Hebbian rule facilitates the formation of a much smaller variety, let's say, of motifs, but two new motifs emerged here, and which I highlighted here in pink. And then we also investigated which motifs emerge through a symmetric rule. And we very interestingly found out that the symmetric rule behaves very similarly to the anti-Hebbian rule and produces the same type of motifs. So again, we could add two new motifs, but again, we couldn't cover the whole set of motifs. So our first conclusion was that a single plasticity rule is not enough to produce all triplet motifs. But just to make sure that this was not due to the very small size of the network, because we are considering only 3 neurons, we also simulated larger networks of excitatory neurons with a single STDP rule. And then again, like they did in experiments, we quantified the motif distributions within each network. But we could observe that again, taking STDP rules from this parameter space, when LTD was much stronger than LTP, again, we could get only the unconnected network, as well as fully connected network when LTP was much stronger, and in between, the same motifs that we would predict for just 3 neurons. So even in larger networks, we, and under, for example, in this case, under Hebbian rules, we cannot get the whole set of motifs. So we moved to heterogeneity of STDP rules, namely combining family of rules. And what we did was to split the network into 3 subgroups, and each subgroup receives connections postsynaptically. So we assign the plasticity rule to the postsynaptic neuron. So for example, the green group will receive connections and also self-connections that undergo a specific rule, and the same for the orange and the purple. In this case, we combined 2 Hebbian rules with an anti-Hebbian rule, and in the bar plot we can see the green, the orange, and the purple show the motifs that emerged within each single subgroup, and they are the same motifs that would emerge if we applied a single rule to the network. But then the interaction of the rule was what was interesting, because it led to the formation of the new motifs that we were lacking with any other single rule. So once we found this way to generate all possible motifs, obviously we wanted to take a step beyond, and we asked whether we could also identify combinations of STDP rules that could reproduce the motifs distributions in real cortical networks as they did in the data. So we used the experimental motif distributions that I mentioned earlier from Song et al. And Turner et al., and we used the same model, so 3 subgroups of neurons and plasticity rules assigned postsynaptically. And what we did was running a large set of Monte Carlo simulations, exploring both the type of kernel and the STDP parameters. So the 3 rules can be all Hebbian, all anti-Hebbian, or all symmetric. But then, or also any mix of those. But also for each rule, we varied again the two quantities, so the ratio between LTP and LTD as shown here, but also the ratio between the time constants. And then we just perform many simulations combining different kernels. This is an example GIF. And then after every simulation, the network evolved to a different motif distribution after plasticity. The goal obviously was to see whether there is an optimal combination of rules and parameters that can reproduce this experimental data. And to quantify this, we compared the motif distributions from the data and the model using the Jensen-Shannon divergence, which is a standard metric to measure similarity between distribution. So here is one example from Song et al., the rat visual cortex dataset, and the model using a specific combination of kernels, so Hebbian, anti-Hebbian, and symmetric rule. And then, as I said, we quantified the Jensen-Shannon divergence, and each point in this heat map represents one simulation, so a specific set of parameters. And the color shows this metric, so the darker the point, the more similar the simulated motif distribution is to the experimental data. And so one example motif distribution emerging from the model is shown here and was a good fit to the data, but I also want to highlight that there is no one single dark point in this scatter plot, but there is a region of dark points, and also this is just one example of all the possible combinations. So this means that we don't need to pick one single precise combination of STDP rules, but different families can produce very similar motif distributions, suggesting that there is degeneracy of STDP rules between STDP rules and motif formation. And then we've performed the same analysis for the other dataset, Turner et al. And again, this is just to show another example. So we also took a different combination, so Hebbian, and anti-Hebbian rule. And again, we see a projection on a 2D projection of this Jensen-Shannon divergence for each simulation. And also in this case, we could find a nice model motif distribution, but also several points in the scatter plot that suggest the degeneracy of STDP rules. And then finally, we moved our analysis to a global scale and found out that our framework can be flexibly applied to also global structures. So in this case, we arbitrarily applied two rules to the network. And instead of applying the rule post-synaptically, we decided to put the rule in a way that depends on either within or across subgroups. So again, we have 3 subgroups. And then by assigning the plasticity rule in these ways, we could find, for example, assemblies or loop or hub networks. This was my final result slide, but as a final slide, I would really like to thank my group, the Georgieva group, who has been very supportive in my work, and especially Christoph, who you all know by now, and Dylan, who will have a poster later, and obviously my supervisor, Juliana, and thank you all for your attention, and obviously I'm open for questions, and I will just leave a summary slide here in case there are questions.
Okay, thank you very much, Claudia. There are a couple of questions, and we can maybe answer one or two. So one question is on the types of neurons. Could you associate these kernels to actually biological types of neurons like PV, SST cells, and so on?
Yeah, so I didn't go into too many details for time constraints, but here I studied motifs between excitatory neurons only. And even though I have an extension in my work where I also involve inhibitory neurons, but I'm still, even if I have inhibition, I still consider motifs between excitatory neurons. But obviously the STDP rule definitely depends on the type of neurons, and the book I mentioned in my references, which is Schuld and Feldman. One second, maybe I can find it. So here there is a nice review of all possible kernels that have been found, depending not only on the neuromodulation, as I was saying, but also on the type of neuron.
Okay, cool. Okay, then one more question. Have you been looking into metastability? So what could you reproduce more of these motifs if the learning rules would change over time? Have you been thinking?
Yeah, basically, the motifs always reach, or like plasticity always leads to a steady state. So basically, changing a learning rule would mean changing the initial conditions. Also, I didn't have time to show like phase planes that I studied analytically, but this is where I can predict the type of motifs that emerge. So, for example, I can say this could emerge or this different motif will emerge depending on initial conditions. And basically, changing learning rule over time would correspond to changing the initial conditions, basically. So I could probably switch from one motif to another motif. So let's say if a learning rule leads to depression, maybe not if I lose all the connections, that I cannot form any more connections. But if the connection gets very low, but then I switch to a very strongly LTP biased learning rule, then I would end up in a LTP all-to-all connected motifs. Sorry.